Hello, I'm Dr. Albert Rizzo, the Chief Medical Officer of the American Lung Association, and welcome to the Respiratory Report. This segment from the American Lung Association at hcplive.com features researchers in the pulmonary field who have received funding from the American Lung Association's Awards and Grants Program. Today's guest is Dr. William Zhang, Assistant Professor of Medicine, Weill Cornell Medicine, and his work is on the abnormal iron homeostasis seen in the alveolar macrophages in patients with COPD, and how that may relate to the pathogenesis of the disease and the exacerbations that occur. Dr. Zhang, thank you for speaking with us today, and please tell us about your work and how you were drawn to this area of research.
Thank you, Dr. Rizzo, for having me. Yeah, this work started when I was a clinical fellow taking care of patients with COPD. And we know that COPD has a tremendous impact on human health. It's the third leading cause of death worldwide. We know that in terms of risk factors for patients, that cigarette smoke exposure is known to trigger COPD development. But many other biological aspects of COPD pathogenesis are not known. And I think in my mind, certainly looking at it from a clinical perspective, it prevents that lack of, that gap in knowledge prevents the development of personalized therapies because we're not able to target really disease mechanisms. And so when I started in the lab, we had first previously shown that taking this is bronchoalveolar lavage fluid from smokers and COPD patients, that there are elevated levels of iron and other iron proteins in the airways of smokers and COPD patients, which we showed using the spiromics cohort that is associated with COPD parameters, including exacerbation frequency. People always ask me what the source of these iron markers are. And my hypothesis has always been that they come from alveolar macrophages or airspace macrophages. And that's been shown in a number of studies in the past. And so we hypothesized that this alveolar macrophage abnormal iron metabolism maybe drives COPD pathogenesis. And specific to my proposal, that we think that the iron accumulation in these very important immune cells in the lung results in their dysfunction and then subsequently an increased risk for infection as well as infection-triggered exacerbations.
So what model are you going to use to further support this hypothesis?
We use predominantly a Murray model of cigarette smoke exposure. And so we first replicated some of the clinical findings from human cohorts that exposing these mice to cigarette smoke-induced iron loading in the alveolar macrophages. And then using various sequencing techniques, we showed that at the transcriptomic as well as proteomic levels, that there's a dysregulation of critical iron metabolic proteins. We dived into that a little bit further to dissect sort of some of our questions, and we utilized single-cell RNA sequencing, and then we found unique subpopulations of alveolar macrophages that have this iron dysregulated signature. And consistent with our hypothesis, this iron signature was accompanied by differential expression of a lot of genes related to alveolar macrophage identity, as well as immune function, suggesting that potentially that there is a connection between iron dysregulation as well as sort of consequences for these cells functionally.
And how do you plan to test this hypothesis further?
Yeah, so when you use omics, it's a wonderful tool to study biology, but a lot of it is still correlational. And so to test this in a causative kind of way, we developed a transgenic mouse model that is depleted in an important iron regular protein. This protein is called NCOA4, a nucleic coactivator 4. Prior studies have shown that NCOA4 has really important roles in sensing iron levels and degrading the iron storage protein ferritin in all kinds of cells, but in particular macrophages. And so we depleted NCOA4 in alveolar macrophages. We showed that by doing that, we can sort of abate the cigarette smoke-induced iron accumulation in these cells. And right now, we're in the process of doing some long-term studies to see whether by doing this to alveolar macrophages, preventing their iron accumulation, that we can sort of deter emphysema development in our mirroring model. One of the major differences in my mind, again, this is coming from the clinician side of my brain, one of the differences between murine models of COPD and human COPD is that we know in human COPD, the progression of disease comes from exacerbations. These are stepwise events, as you know, often infection related. And while murine models were not really able to fully recapitulate that, we try to mimic this. And so we also had started experiments where we exposed mice to relatively shorter periods of smoke exposure, and then giving them a pathogen such as Streptococcus pneumoniae to sort of mimic a second hit model that recapitulates human exacerbation. Our animals initially seem to have a phenotype of survival from the second hit of Streptococcus pneumoniae. With more replicate experiments, it's not clear that they're completely protected and that there may be a double edgedness to sort of deleting such an important iron regulatory protein in these cells. Obviously, sort of our hypothesis started from alveolar macrophages accumulating iron and potentially causing dysfunction. But to get rid of an important protein that breaks down ferritin and allows the cell to retrieve iron when it needs to, especially when macrophages are fighting infection, I think probably also hampers them to some extent in this infection model. And so I think that the biology behind this is more complex than we initially hypothesized, and and not so straightforward.
Understand. So I understand this work has been presented at the American Thoracic Society International Conference. What are some of your next steps?
We're completing, we're completing almost all the experiments in the murine component of this project, and we're preparing our manuscript to be submitted and hopefully published later this year. We've the focus of my laboratory certainly is studying COPD as well as alveolar macrophage function, and so we're trying to stay active during this time. And during the funding period, with support from the American Lung Association, we were able to publish additional manuscripts that really centers on the effect of cigarette smoke on alveolar macrophage inflammatory response. We're also trying to translate some of our findings back to the bedside. And so we had already published a proof of concept study that we could use deferriprone, an iron chelator, to reduce intracellular iron levels in alveolar macrophages obtained from smokers, but interestingly not from subjects with COPD. And so that was, again, an exploratory study, whether this difference between smoker macrophages versus COPD macrophages, whether there's a biological difference that explains what we found, or whether it's just due to inadequate dosing or something like that. That is the subject of our ongoing studies. We're also trying to finalize a manuscript where we evaluate alveolar macrophages obtained from human bronchoscopy in smokers and COPD subjects in the spiromics cohort, and trying to find specific RNA signatures in human transcriptomic studies that sort of mimics the findings that we had in our RNA-seq and single-cell RNA-seq studies in our murine model. And that work has been presented at the ATS already, and also should be ready for submission later this year.
Very interesting research. So I want to thank you for your time today and your work in this important area of COPD, and particularly the exacerbation relationship. Good luck in your ongoing projects.
Thank you so much for having me. As I've said multiple times, as young investigators, these kind of support from the American Lung Association helps us bridge from one grant to the next. And so all of this support is incredibly valuable to all of us. And I wanted to thank everyone here.